Good evening, everyone. I hope I'm audible.
Yes, Sumati.
Thank you. Let's wait for 5 minutes.
Swarupa.
Ma'am? Ma'am, please tell me, ma'am.
Yeah, you are the host, Amma. You just take care of the meeting. Okay?
Okay, ma'am. Okay, ma'am. Sure, ma'am.
Okay. Right, I'm in the journey and I don't know whether my signal will be will be there for me or not. You just take care. Huh?
Okay, ma'am.
Now, one more thing, there's a YouTube link is provided
Yes, ma'am, we got it, ma'am. Yeah, it is already telecasting in YouTube, ma'am. It is on live now.
Hello everyone.
Good evening, sir.
Hello, sir.
Hi.
Yes, sir. Yes, sir. You are audible and also visible.
Okay, let me know when to start it so that I can present my screen and
Sure, sir. Sure, sir.
I think this screen sharing has been disabled. Maybe for a group or something. Please do that.
Hello, sir. Dr. Prasad Garu. Good evening. This is Sayana speaking.
Yeah, good evening, sir. How are you?
Fine, sir. How about you, sir? Are you feeling well?
Yeah, fine.
Yeah, thank you, sir. I'm online. I want to listen your class, sir.
Oh, great of you.
Thank you. It is almost 5:15, sir.
Yeah, just I'm waiting from host side. Once they said more than 80% people are joined, then we'll start presenting.
Okay, this information has been given to many Navodaya students also. They may join. And I informed to Nandakishore and many students are willing to listen your lesson online,
Oh,
sir.
great. So,
JNV students may also join. And thank you, sir. I will be online.
oh, great.
Thank you.
Hello, Swarupa ma'am. Can you just give me co-host? I'm unable to share.
Sumati, now you're co-host. Please share.
Okay, right. Just having some problem. Okay, good evening, everyone. Sorry for the 5-minute delay. So just waiting for some more participants to join. So meanwhile, a very warm evening to everyone. So I'm Dr. Sumati Natarajan from the Department of Microbiology from Telangana Social Welfare Residential Degree and PG College for Women, Mahendrahills. So I cordially invite everyone for this national webinar on the past, present and future of fermentation. So this, especially on a day like the International Microorganism Day. So this celebrations, we have been doing it from the 14th. So we recently also had a microbiology art competition that was conducted so that the students are made aware of this day. And we all know that microorganisms are ubiquitous and they are making their mark in every field and we cannot survive without them. At the same time, these invisible creatures can very well wipe the mankind, right? So the corona pandemic has made the world realize the importance of microorganisms, the havoc that they can cause, and at the same time, the huge potential that they have for human benefit. So microorganisms make their presence felt as they are present in huge numbers on any other life form that we see. So we are largely dependent on them, be it our kitchen, industry, healthcare, and also the functioning of our very own body. So they are crucial in even maintaining the ecological balance, be it any ecosystem. Also, they are now being seen as the first creatures to visit the planet Mars. And they can also control your thinking. That is something new that they are doing as psychobionts, right? So here, International Microorganism Day is celebrated to give credit to these varied microforms and also spread their awareness among the general public. So here, actually, this day has importance because 17th September 1683, this was the day when Anthony von Leeuwenhoek, the father of microbiology, the discoverer of the microforms, actually wrote a letter. He penned a letter depicting all the drawings of the microforms to the Royal Society of London. So it was on this very date, 17th September. So hence, this is the date it is being celebrated as International Microorganism Day. So I welcome you all again, and we are joined by few dignitaries today. So I welcome Professor Arvind Deshmukh, who is the President of Microbiology Society India, Dr. G. Nirupa, AZU and Principal of our college, Dr. Rajni Paspuleti, the Zonal Coordinator South Zone, TSWRDCs, Dr. Prasad Arnala, who is our Chief Guest Speaker. He is the Senior Scientist, Fermentation Technology at Dr. Reddy's Laboratory. So I welcome everyone once again. I now invite Dr. Swarupa Rani, Head, Department of Microbiology, to share her thoughts on the occasion and also introduce our speaker for the webinar. So as Principal Ma'am is having some problem with her, internet and speaker, so I just hand over the session to Swarupa Ma'am, who is the head of our department. Over to you, Ma'am. Ma'am, we are unable to hear you.
Unmute yourself, ma'am.
Yeah, thank you, Sumati. Very warm greetings to everyone. I wish you all a happy International Microorganism Day. So this day aims to raise awareness about the importance of microorganisms and microbiology research among the general public. Microorganisms, invisible to the human eye, they are everywhere in, on, and around us. Microorganisms have made headlines in 2020 for all the wrong reasons. Despite their negative effects, they are an integral part of our lives and have hidden power in many everyday activities, serving to ferment foods and treat sewage to produce fuel, enzymes, and other bioactive compounds, and a vital components of fertile soil. In the human body, they make up the human microbiota, including the essential gut flora. The knowledge of microbial literacy is vital to know how they impact our lives and how they may be harnessed for the benefit of humankind. This International Microorganism Day gives us an opportunity to promote the positive contributions of microorganisms in human health, culture, and throughout our daily lives. On this occasion of our department, in association with Microbiology Society of India, has organized a microbiology art competition from 14th to 16th September. The president of MSI, Professor A.M. Deshmukh, sir, has been very active and supportive in our initiatives. Today's webinar is on a very important process, which is fundamental to most microorganisms and can be related to our survival, that is fermentation. Fermentation is the key to economy, be it health sector, food and beverages, waste treatment, or even in your kitchen. With these few words, I would now like to introduce our speaker for today's, Dr. Prasad Ernala. Sir is senior scientist and fermentation technology specialist at Dr. Reddy's Laboratories, Hyderabad. Initially, after obtaining his master's degree in microbiology in 2008, he joined AML industry, where he carried out both basic and applied work in microbial fermentation technology and developed the industrial process for production of vitamin C. He worked in a few projects related to production of curcumin conjugates at the Indian Institute of Chemical Technology. He successfully completed his PhD under the supervision of Professor Gopal Reddy sir, Osmania University, which dealt with biosynthesis of glucosyl curcumin and glucosyl tetrahydrocurcumin. To his credit, he has one patent and 7 papers published in various international journals. And also, he's also a very good artist from the Department of Microbiology, Osmania University. I welcome you, sir, for this webinar, and the session is all yours, sir.
Thank you, Swarupa madam, and great of you for the good words about me and nice graphics. So, just before presenting my screen, I would like to thank Sir Ronald Rose, sir, Secretary for the Society, and Dr. Praveen sir, Joint Secretary, Dr. Rajni madam, and Dr. Nirupama madam, Principal for the school, and as obviously you, Swarupalani madam, HOD, and as well as our friends like Dr. Smriti and Dr. Vasavi madam. I would also extend my special thanks to Deshmukh sir, who is President for Indian Microbiology Society. So, let me share my screen and start my presentation. So I will also hope my screen is visible to everyone.
Yes, sir. Yes, sir.
So, first of all, I won't neglect the students of the call who specifically gave an opportunity to me to share my point on the topic, like past, present, and future of the fermentation. I won't call them as student anymore because already they are graduates, and hopefully they are future scientists who are going to enter into the science and explore more than what we are doing at present. So, happy congratulations to the all science, microbiology science future scientists who are coming up from the academics. So let me start my presentation. So, as we are presenting it to the future scientists, I want to make clear distinction between the past and present science. Most of the academics, they will keep on saying the history of whatever the science it may be. The history always says we did this from these many years. It has been under practice. But as a future scientist, you just note down the history facts, but don't by heart keep history in the mind. Just look into your dreams and look beyond the history. Look future of the science, whatever the science you are reading, and make that dreams dreams about the science into true. So if we comes to fermentation, major events in the history, it has been if you see a table and the picture, there was contradictory in the years itself. So we may not celebrating any birthdays for cheese innovation, wine innovation. So don't deep dive into these specific dates, whether it is 6000 BC or 7000 BC. Somewhere in between, it has been invented and it has been practiced. Like cheese production was done in Iraq in 7000 BC, and so on. Wine making, it is in 6000 BC, and yeast preparatives discovered in Egypt in 4000 BC. So all those things are it was when you go into the academic books or Google it about the fermentation. Many a times you just find about fermentation of food sources. So fermentation now it has been evolved to a great extent. No more the food fermentation is just a fermentation. So I can say, even a few of my friends who are working in chemistry and other areas of the science, when they say I'm starting a fermentation batch today, means they will just simply say in Telugu, tode sunnava. It means in our Telugu, we are adding inoculum to the milk, inoculum of carb to the milk. So the fermentation term itself, it has been coined with the very primitive way of food making, food fermentation. So all the history revolves around the food fermentation only. That's why up to this level. When Pasteur came and started producing antibiotics and a few other biomolecules through the microbial source, then it started evolving beyond food to something else called life-saving drugs. So it is in 1953 AD where Nicene marketed in England, and since approved to use as over 48 countries. Then onwards, the biomolecule of Nicene, which got into the picture, which was produced from the fermentative source. So again, when we come, our title comes as past and present of fermentation. Let us discuss about black and white part of the fermentation, which is early fermentation. So where if you can go through the headings, all are revolving around the food, like alcoholic fermentation, acetic acid fermentation, lactic acid fermentation. They are very much relevant with the food fermentations. When you comes to the color side of the fermentation, current scenario of the fermentation, there are too many, a wider range of applications in the options of fermentation to explore into the market, industries, enzyme productions, metabolites, RDNA technology, which is very new to the, which may not be very current, but the technology which at present ruling the industry is RDNA technology, and beyond the RDNA technology, transformation products, the products which are future for the future economic growth, biofuel products, and even the products which were needed for the bioremediation, like waste management and aromas, and even nowadays, textile industry also depending on the bio dyes, dyes which were produced from the fermentation. So if we keep on exploring the microorganisms, they will keep on extending their help to mankind in producing the products what people are expecting. It may be like a lot negative done by this recent pandemic by corona, but as a microbiologist, we should thank a bit to the corona. Like most of the world population came to know the importance of microorganisms as well. Like what is the microorganisms and importance of microbiologists as well. I think most of the people sitting around accept my words. And let me introduce a bit about the science of fermentation. Here, how this fermentation goes. Right side, if you can see, exactly the fermenter, industrial fermenter, how it looks like. So, at present, how we are, with what we are working on. And this left side, it is a lab scale fermenter where we use substrates, very low price wise, like cheap substrates to produce the highly valuable products. We call it as pharmaceutical ingredients, active pharmaceutical ingredients. So we will take substrate like sugars, nitrogen sources like molasses, all those things, and the concept of taking sugars and nitrogen sources. So let me give you a small introduction to this. So at any point of the time, if we want to prepare a curry, we will choose the ingredients like haldi, oil, chilies, onions, vegetables, everything. So altogether at the end, it will become a curry. So if we want to generate, we want to microorganism. So fermentation is nothing but applying microorganisms and extracting the products from the microorganisms. So though microorganisms are living organisms, but the content of content of microorganisms is nothing but the carbon and nitrogen. If we can dissect stage by stage, at the end we will move to DNA, RNA, all those things. So into the cellular parts. What all these cellular parts consist of? It is nothing but elemental nitrogen and elemental carbon finally. So that we are adding this elemental nitrogen and carbon from different sources like sugars and molasses, soybean, if it is some other applications, animal source of nitrogen. All those things we will feed into the fermenter, and we will add some mother inoculum. So if we give one or two bacteria into that nitrogen source, this bacteria starts utilizing elemental nitrogen, carbon, all those things, whatever available, including gases and other things. They will start multiplying, and the two are, I mean to say, numerically, I am saying 2 or 3, so, but the inoculum starts multiplying, and in the course of time, maybe in a couple of hours, it will start getting turbid. It means they are multiplied vigorously, and 2 become 2000 or 2000 become 2 million. So it's gone. So once the population of bacteria is increasing, their need will be increased, and we keep on supply to the based on our product. And once it saturates our at the point where bacteria starts producing our desired product, we will start collecting from the fermenter, or it may be sometimes like a batch fermenter. We will still wait till the saturation point of product formation, and we will harvest everything, and we will extract our product from the cells. Maybe they are extracellular, intracellular, based on the things. So based on the things, if it is extracellular, we need not to kill, or we need not to manipulate cells. We just separate out cells from the media, and our product would be in media. If it is intracellular or superficial, we have to extract, we have to separate out liquid portion, and we have to separate out the cellular biomass, and Maybe if it is intracellular, we have to kill all the microorganisms and extract it from their cell debris. That is another part of the extraction. So if it is superficial, we have to add some unbinding buffers or solvents, whatever. So it may be like suitable for the product and cellular structures, and we have to get the products into that buffer. So like these are a few examples which I gave, like cannabinoids and all those like active pharmaceutical ingredients what we are playing with right now. So like what is the application? Like finally, like what we want to do it with this type of chemical ingredients. So finally, like as a pharmaceutical scientist, I'm working with so many like drugs like anti-cancer, anti-malarial, antimicrobial, antiviral, anti-diabetic, so many like so many other applications specifically for pharma industry. Apart from pharma industry, there are other applications like energy production, all those things. They may come in like upcoming slides. So this is how like fermentation process works. So it may not be always yeast or bacteria. So we can explore many like almost all the microbial sources like bacteria in bacteria itself, like E. coli, Pseudomonas, Bacillus, everything based on the product specificity, like and organism specificity, we will choose the bacteria. And I can say like in like recent trends, like obviously like E. coli became the like a friend of humankind, like who are working for fermentation, because the handling and all those things were very like it is comfortable for us to manipulate E. coli maybe like by using recombination technologies. So whatnot, we can produce almost all the molecules which are produced by any living organisms. Like, so the only thing like you have to trigger out which specific gene is coding this that biomolecule or whatever the chemical molecule which is which need to be produced. Then we will simply introduce that gene into the host cell, like a selected host cell, and we will start like inducing that gene to be multiplied, and later on the specific product will be produced by the fermentation methodologies. So here on like may I ask like do you have any questions from this slide or from the things what I said about the fermentation? Let me know, like instead of like one-way traffic, let me I want to take questions slide by slide. Or like you can post it in chat box. I will answer one by one, maybe at the end of presentation. Anything is okay. So let me like move on. So the initial like when this fermentation technology is wrapping up globally, and like there are so many products coming into the market other than the natural like products for the fermentation, like this isobutanol is one of the richest product produced from the fermentation technology, and the product itself has such a wider applications. No, like it became the like initial in the initial days it became the like king of fermentation technology, like in plastic industry, rubber industry, pharmaceuticals, even agriculture. So in pesticide industry, like they used this product as one of the supplement, and other than that, paint industry, like isobutanol itself is used as one of the solvent for the chemical extractions. And in polar countries, it was mixed in with the like fuel, petrol and diesel, so that the freezing point would be like maintained in that states. And even nowadays, like isobutanol is highly purified and is used as jet fuel also. So this is one of the product which entered into the market and ruled everywhere. I mean to say like all the sectors of economy. So let me say other than the pharmacy and other sectors, there are other industries where this fermentation technology can be implemented. In textile industry, people are using it for dye production, like color production. Even specifically this actinomycetes, which were where the soil is good source of that actinomycetes bacteria. Though almost 80 to 90% of all the antibiotics are produced from these source of actinomycetes bacteria, the same bacteria can also be utilized for the production of dyes. And even in current trends are current fermentation trends, the dye is a byproduct of any antibiotic producing fermentation process. So as fermentation keep on evolving with the technologies available globally, so this type of byproduct scouting always helps in reduction of cost that is applicable for the fermentation production. Here I can say why industry, why specifically fermentation industry is getting low in comparison with other industries like pharmaceutical, automobile, even almost all industries. So here, as a microbiologist, you should focus, like all our upcoming young scientists should focus, taking a fermentation unit, installing a fermentation unit and operating it. Need to have a huge cost included in this, instruments wise and operation wise, energy that is required for the operation. That is one of the reason why these people are afraid to take fermentation industry as a good source of finance generation. But obviously, in future, upcoming future, I just maybe 2 years back, one of my professor who met me in one conference asked me what is the future of microbiology, or what is the future of fermentation in upcoming years. At that time, I said it would be going to another organic chemistry, a decade or 2 decades back, when the organic chemistry started and pharmaceutical industries growing up with a vigorous manner. So at that time, every organic chemist got good positions in industry, and organic chemist was earning equivalent to the IT people. All those things were happening. But in recent trends, no, it was microbiology and fermentation industry which is acquiring that position of organic chemistry. And I'm not saying organic chemistry may go down, but obviously fermentation will come up beyond the organic chemistries, because globally the green chemistry trends are coming up, and use of hazardous chemicals, use of hazardous solvents, and all those things has to be minimized, it is cautioned by UNO and other organizations globally. So alternatively, if the green chemistry is nothing but they have to work on enzymes or alternative methods of producing pharmaceutical ingredients, which which has great scope for the fermentation industry. So people who are they are studying microbiology should not get discouraged. So future will be there for any science. If you are a student and taking science as a career option, definitely do your best and learn science to its core, just not for the exams, but it's learn everything, every bit of the science. Just you will do the things well, like you will keep the things in place. So like let me move on. Like fermentation is also placed in automobile industry, where ethanol production in Brazil has the greatest impact on economy, and the ethanol which is produced from like agriculture source has like moved on to the automobile industry, where it was mixed with fuel, and the part of ethanol which is mixed is in a major quantity, and which is obviously produced from a lower and cheaper quality of substrate. Obviously, like it added value to the economy. And again, like recently, few people might have heard about like synthetic meat, and I can't say synthetic, it is bio, like fermentative product, and even milk also, like not from original source of cow or buffalo, but like milk also, like which was produced from microorganisms. Like it may not be like a directly complete milk, but like all the nutrients which are present in the milk will be produced from fermentation, and they are mixed in appropriate ratios, and will be added with other supplements, minerals or vitamins, I can say. So like it can be a supplement of milk. So like microorganisms which were generated by recombinant technologies, giving food sources, like in during our primary school, I can say primary school or initial college days, we used to say like population is alarming, like population is growing in geometric way, whereas resources are very low. Like I can say like the land may not grow, like the production of food producing strategies has to be altered, and like cropping things will be altered. So this is one of the good like best thing that fermentation give to the mankind. Like we are producing food in vessels, not on land, or not on like the field. So this is one of the like greatest thing in recent times we come across from the fermentation industry. So the other thing is energy. Like though it is a classical, but things are moving to favor fermentation industry to produce energy as well, like biomass utilization, like garbage utilization to generate energy is becoming economically friendly to the mankind. So the in future, like people may purchase garbage from our gardens and households. So that time may come, like instead of paying bills for the energy, we have to we may get credits back to our utilization of other things. So this is one of other application that fermentation has. So let me revise a few slides what we touched upon about the fermentation things, the global fermentation market, and by its application. So food and beverages, where fermentation is applied, and pharmaceutical industry, as well as agriculture, where like pesticide, biofertilizers, and biopesticides has been produced from the fermentation industry, and personal care products, like beauty products can also be produced from this fermentation technology. Textile and leather, where in leather industry they use different lipase and other enzymes can be produced from this fermentation, and in textile, like as I said, dyes. And apart from this, other applications like this biomass degradation and energy production. So right side are the captains in industry who are implementing fermentation as their major activity. So all these companies globally acting as fermentation leaders, maybe like in future people can explore their jobs in these companies to implement their ideas in fermentation. So let me move on. It is the time to expect a few things from the future scientists who are going to enter in fermentation. So maybe degree students may not understand all the topics, but keep in mind what we have to do in future, what we have to crack in fermentation industry. So if there is a hurdle or if there is a question before you, then only your mind will activate and your mind will thought of how we can crack this question or how we can break this hurdle. So let me post a few questions from the classical current fermentation industry and what can be done in future fermentation, so that what you can add to the future fermentation industry. So let me though the slide is a big texty and has so many things to discuss, but let me read a quick thing. So microbial classes. At present, we are using E. coli, Pseudomonas species, Corynebacterium, Glutamicum, Aspergillus, all those things. So, why this can be replaced, or why is it okay to replace? Why it is the need for replacement? I can say all these are stringent at their requirement. Suppose they will grow at specific pH, they need specific substrates to grow, and they need sophisticated atmosphere. They need a good amount of raw materials, good amount of food to grow. All those things, to supplement all these things, we have to spend a lot, we have to spend time, money, all those things, energy. So to avoid that, we need this microorganisms which can extend their help to humankind, irrespective of by extending their expanding their boundaries, extremophiles, they should grow at different temperature, either 5 degrees to maybe around 60-70 degrees, but parallelly they have to give same productivity. So that is one concern, and they should be feed upon very low cost, or if it is gases, instead of sugar as a carbon source, they should absorb carbon dioxide, carbon monoxide, or other pollutants, carbon pollutants which were released freely into the atmosphere. If they can utilize these type of carbon sources instead of sugars, it would be another asset for the fermentation industry. Then sterilization. Yeah, first of all, the sterilization at present consumes a lot of energy. You have to sterilize multiple times. It needs a huge amount of energy for heating, cooling, and all those things. So it may be required, but the amount of the extent of sterilization can be reduced. It should not get that easily. The organism which we selected for our purpose should not get contaminated that easily. So that is another area where upcoming fermentation scientists can explore. And the next point is part of this discussion, contamination. So at present, contamination issue in fermentation is a big devil for the fermentation scientist like me. And if we can control this contamination without utilizing so much of energy, it would be a added advantage. And obviously, freshwater utilization in fermentation is very high. So instead of freshwater, if we can if our organism can adapt for the seawater, it makes good value for the thing. So they can the upcoming future fermentation technologies can explore this part. And growth process. At present, most of the industries which were taking this fermentation as a part of their production. So they mostly use batch and fed batch fermentations. Very rarely people are using continuous, but continuous fermentation, if they keep on focus on developing products on continuous fermentation, they can reduce a few of the above points, like they can reduce frequent sterilizations and use of much more energy for the sterilization. Even they can reduce the contamination chances. Like once the organism is grown, means there is less chance of contamination. So if this continuous fermentation acts as good source of production, then they can reduce few things which were discussed above. And substrates, obviously, at present we are using glucose as carbon source, and soya tone or peptone as, which are pretty much food sources of humankind as well. So we are giving cages of glucose to the bacteria to produce our product, whereas the glucose can also be given to the people as well, directly it can be utilizable. Instead of that, if microorganisms have to be adapted for complex carbon sources like starch, gases, and mixed substrates, even I think Dr. Swaruparani might have seen many of the substrate in her PhD. Her group used to use so much of this grass and other cellulosic materials for the production. So if such substrates comes in place for the industrial scale production also, it would be another asset. And products. At present, most of the fermentation focus on single product. If we are focusing on any one of the antibiotic, we will simply focus on if something forms in lesser than 10% of quantity, we will eliminate it as impurity in the final product, and we will focus on the single product. If we can focus on if we can develop a bacteria which can produce multiple products, like suppose one enzyme which is produced by specific bacteria as an extracellular, which can be extracted into the liquid part of the fermentation, and another product which may be a peptide or some a drug molecule which can be produced within the cell, like intracellular, can be it will be easy for get two products, and it will be good for getting two products in a different state of fermentation. That is another thing where we can explore fermentation technology. And sulfur and phosphate ratio in fermentation, it is reasonable at present, and it can be extended to a better ratio for the elemental balance. And wastewater wastewater. After using this fresh water in the fermentation, at the end of fermentation, we will drain out 1,000. If it is 1,000 liter fermentation, we will drain out more than 1,000 liter of wastewater from the batch. So that is another concern where water ambassadors are scouting for the thing, you reduce the utilization of fresh water and look for the alternative, or reduce the wastewater draining out from the fermentation industry, and go for recycling the water, and all those things coming into the place. So it may be it may be another criteria to choose to develop a fermentation industry. So all these things are let me discuss a few more things. But why I'm discussing these things means people from different universities or different areas of globe keeps on asking in different social media, sir, what can be done in fermentation research? Suggest me one topic so that I can choose and I can explore in fermentation technologies. So all these are the research areas itself. If they can focus on keenly and they can get the things done, every point can be one research area for the academy. Like I can't say that just academy, but any scientist who are working in fermentation, every point has its own specific area of research. They can explore as much as they can, like taking fermentation as one of the research area. So like downstream process, in fermentation area, downstream is one of the critical and key element where people has to explore a lot. Like different microorganisms has a different tendency to produce the product in different part of the formation. Like suppose if it is gases, like recently I can share my experience. Like I didn't do, but I came to know, like through the peers working in the industry, they are producing gasoline by the fermentation, which is a gaseous product. They have to isolate gas from the fermentation, and subsequently they have to supplement the nutrients back into the fermenter. They devised a technique using TFF, tangential flow filtration, and it was they got patented it, and it is like actually both are different equipments which were used in fermentation technology itself. One is fermenter is in upstream, and TFF, tangential flow filtration, in downstream. So they merged both the equipment, and they kind of like a different mechanical instrument itself. It was like a gift for that gasoline industry. So like that aeration, at present the microorganisms are industrial microorganisms which were used are consuming a lot of oxygen. In fact, if one can see my LinkedIn profile, like in LinkedIn post, at the time of this pandemic, I posted one regarding this. Like I asked the fellow fermentation scientists to stop taking batches with E. coli. Like as E. coli is consuming so much of oxygen, we for even small scale fermenters, we use a couple of oxygen cylinders to run E. coli batch for 24 hours. So at the time, the oxygen cylinders were not available in the market. So we it is very essential for hospitals. So like that was the one of the concern where we need to focus to narrow down the oxygen consumption in fermentation industry, so that alternative microorganisms or alternative strains can be developed. So next is the pH. pH is one of the concern in fermentation technology, where we will play with small variation in pH indicates either there may be like nutrient shortage or product formation, something like it. On the back of it, it is it is a negative effect for the bacteria which is growing inside. So immediately we need to add, and we need to supplement something to manage this pH. So that should not be done. Like if fermentation has to be robust, the organism should have a wider range of pH, as well as other physical factors like temperature, osmotic pressure, all those things. So another concern is bioreactor, where that's what in my initial presentation I was mentioning. It takes a huge cost financial wise to start a fermentation industry. So as it is like a sterile specific, a chemist can do his reaction in small glass flask, whereas fermentation scientists can't take that chance to it in some open vessel. It has to be very sterile, and all those the physical parameters has to be met. So automation, aggregation, induction, cell lysis, and other well-educated labor. So in few chemical industries, they will just take the SOPs from the scientist, and they operate. It will be operated by normal people also. Whereas in fermentation, technology, like each and every parameter is crucial. Fermentation industry can't afford to lose any of it, so they need well-educated scientists in place of labor. This is the thing. So like I think people may get bored with this slide because so much to discuss and so much to focus on it. I don't know like how much our young scientists observe from this slide, but just I think I will share with Swarparani madam for further discussion, and if any point of the time, if you have anything to discuss about the things present in this slide, I am open to discuss at like either personally or through the mails, wherever you need. So just why recombinant technology is a bit slow. So recombinant technology started long back, like when it was multiple decades back, but still it is not coming up to the level that it should come. So because now like since this analogy of fermentation research started, the inquiry, the quality of products has been parameterized by the chemical and sensory point of view. Like what is the chemical? Is it acid, base, nutrient, single cell protein? Those things were important. Where it is wine or beer, something like that. Where in the current scenario, it evolved to a level of structural elucidation and the composition of it by different industries. So like as you can see from the picture, like mixed bacteria, like when primitive age of fermentation, there was no one has the knowledge of which bacteria exactly it is producing wine, or which bacteria which exactly it is curdling the milk. So they used to add something either knowingly or unknowingly, and they used to get the product, and there was no safety or shelf life of that product. If the consumer accepts it as a good, it was marketed. Whereas later on it moved to defined strains, like when it has perfect science in place for science of fermentation, they chosen the specific, like suppose if it is yeast produces duff and yeast produces ethanol, all those things, then they specifically isolated that specific bacteria or microorganism and used it for that specific purpose. And at that point of time, consumer was appreciated and accepted the products. When it further evolved into the specific technology, defined the strains, like within the equally, which strain has to be used. Within the actinomyces, which strain has to be used. There are 1,000 lakhs of strains in every species. So that strain specificity came in the recent trends, and that specific strain will produce the specific product which related to the, I can say, pharmaceutical industry. And back of that, there are legislation like law in every country, which is focusing on how much, what is the exact strain, what is the genetic code of that strain, total gene sequence of that strain, whether it is matching with the standards of industry. Is there any other gene which is expressing along with which can cause damage, like which can impregnate through the sources, and it may cause damage to the society in a long run. So all those things came into place, like RCGM, all those things came into place. In every country has their own specific laws to monitor this recombinant technology. So these are few things which are blocking recombinant technology. Like I can't say blocking, but pulling back or making recombinant technology stringent, pass through the bottleneck of these rules. So combination of different technologies that are supporting to the fermentation industry. Like in this slide, I'm going to present like how we will select organisms like host cells, whether we have to go with E. coli, whether we have to go with yeast or some other organism, bacillus, other what makes the best choice of this specific product based on the host cell genome and the genome which is meant for the production of a specific product. Once the host cell is selected, we will check the pathway, like what is the pathway, like the final product can be produced. And the process, finally, the process, once the clone is developed, we will go for process optimization, physical parameters, chemical parameters, and biological parameters, which has to be optimized. And finally, the process will be scaled up to the industrial scale where production can be done. And these are like different parameters what I mentioned in the previous slide, which can be monitored either parallelly along with the fermenter, or we have to take sample outside and we have to monitor the parameter. Like all the green parameters can be monitored parallelly with the fermenter, with the using real-time technology, where like a DO sensor will be there, like a dissolved oxygen sensor, pH sensor, like optical density can be calculated based on the spectrophotometric methods, and temperature has its own temperature probes for the measuring. And apart from that, foam detection, chemical composition of media ingredients, what we are taking, vessel pressure, air and gases, specific gases like oxygen, nitrogen, carbon monoxide, carbon dioxide. All those can be measured with using real-time technology. Whereas other parameters like carbon sources, nitrogen sources, and mass like weight, cell weight, all those things have to be measured by taking outside the fermenter. So this is the, and in future, like in future fermentation technology, as I discussed few of the points in the slide what extensively discussed. So like pulsing electricity into the fermentation broth, few like mild electricity into fermentation broth increases enzymatic activity, which is which directly or indirectly influence of final yields of the fermentation, and improves fermentation time, as well as like instead of taking it for 10 days, we can conclude it in within 10, like 5 or like lesser than the what is required. Enhance the membrane permeability, like suppose the product is within the cell and we want to get this product into the like outside the cell so that it can be extracted parallelly. So by giving these pulses of electricity, we can reduce the we can enhance the membrane permeability, and product can be extracted outside, like without harming that specific bacterial cell life. And few like even cold plasma beams, like light beams, can also act similarly, as well as like ultrasonic sounds can do the purpose. So these by using these things, we can solve few of the we can even detoxify the products. Like suppose if it is enzymes which can be used for the food, which can be used in food industry, where the mycotoxins are key players which damage the food products. So to reduce that mycotoxins, these light and ultrasonic waves can be used to break down that mycotoxin proteins and make the product toxin free. So this is the slide which elaborately says micro agriculture, like production of meat and other food grains or other products by fermentation industry. So this is once again the recap of a technology called cellular agriculture. were initiated in 1833. Using the proteins are first described by the Dutch chemist Gordon Johnson Smolder, and when it passed on to 1982, again 1930, 1918. Recently, the fermentation products of enzymes are ubiquitous in food and household, and it is going on. At present, it is 1920. Like 2021, and the history of fermentation moves on. So the fermentation products can be implemented at any industry, like either cosmetics, energy, food, and all those things. That's what I'm saying. Like fermentation industry will become another organic chemistry. So on the future of fermentation, microbial products and chemicals, minerals, biofuels, food, what all we discussed can be get by the fermentation technology. The modern bioeconomy consists of biotechnology. Here, I want to stress one thing: how this technology evolving. Why this fermentation? Whereas during the past time itself, or something in between 80s in 70s. Why this technology have not been developed, either recombinant technology, or what can what is the backbone of evolving technology, either it is biotechnology or microbial fermentation technology, whatever. So once the availability of sources, availability of, suppose after invention of microscopy, only one can see the microorganism, and they can play with the microorganisms. They can utilize the microorganisms for the different purpose. Like for every technology has to be developed, there should be parallelly other technology has to support that technology. So if fermentation technology has to evolve further, so along with from biological microbiology scientists, other scientists like electronics, mechanical, whatever, other physics scientists, biotechnology scientists, whatever, all has to work parallelly. Like if someone from electronics scientist can develop a sensor carbon dioxide sensor or some sensors, then we can use that sensors in our real-time study, and we can implement that use of that sensors in our fermentation technology. So the technological sciences should go parallelly, and one by one, the technology will be grown parallelly. That's one thing. By the technology, recent technologies are getting evolved day by day. Just previously, if one has to develop one new technology, would have taken them suppose 10 years or 5 years. Nowadays, we can get the technology developed within months or maybe within days. That much evolved the sciences are. So on the future fermentation, this is one, nutrients can be recycled or reused for the fermentation. Changing morphology of the self aggregations of bacteria can be studied, and extracellular products and intracellular products can parallelly be produced. And use of ion exchange, ion exchange were previously used in chemical industry, but the ion exchange resins are pretty much useful for the fermentation product extraction. So all these technologies can be helpful for the production of pharmaceutical products, and they make our work easy. They make fermentation scientists work easy, and we can give the best products to the market. So this is the global fermentation product market area wise and quantity wise, whereas our Asia Pacific countries are equally competing with North American countries, and both are leaders in the market at present, and maybe in future, we may lead the places. all other places of the world. So maybe like India and China together may lead all other countries in coming future. So let me go for question and answers from my side. These are two questions, as it is Day of Microorganisms. I should wish all the microbiologists happy Microorganisms Day. And let me tell you, as we know, most of the species, most of the eukaryotic species are extinct. They can undergo extinction. That's what endangered species are getting identified. Do you think bacteria can also go extinct? Is it possible? As the multiplication time of bacteria would be within hours. So if one bacteria survives, it can generate hundreds of bacteria within a day. But whether is it possible extinction of the bacteria? If someone says answer at the end of question and answer hour, right? It's good. I gave a clue. New research says yes, it will undergo extinction, but how? And second question is, do microbes produce anything that can be produced by living organisms? If so, what is the chances? How it can be? So let me 3, 4, 5. I'm expecting from your side.
Yeah, thank you, sir. I hope I'm audible.
Yeah, thank you all, and let me know what all can be discussed.
Yes, so it was a very wholesome webinar on fermentation and the technological updates in it. So just a few questions are there in the chat box, like how industrial production of biofermentation model may help in our human digestive system if we introduce fermentation. Is this possible process, sir? There are few in the chat box.
Yeah, I'm going through it. Just a second. Industrial production of biofermentation model may help in our system if we introduce fermentation. Is this process possible? Actually, I didn't clearly understand question. How industrial production of biofermentation model may help in our human digestive system? Yeah, I can say every bacteria. If I will give one funny example, if everyone might have seen Robo movie, Robo 1 or Robo 2, Rajinikanth movie. So every robo has the capacity to produce generate other robo. Robo will generate other robo. Similarly, every microorganism has a capacity to produce other microorganism, as well as every microorganism itself has a we can consider microorganism as a micro fermenter. So where the within that cell, it has all the machinery to generate product, as well as itself required nutrient, I can say vitamins or whatever the micro elements which are needed for the multiplication, growth and multiplication both. So it is self-surviving, utilizing nutrients available from the external environment, and as well as it is giving product outside, or it is generating product as well. So you can say a microorganism itself is a fermenter. We need not to show an industrial fermenter to the people. It is the fermenter which is monitored, which is operated by motor or controlled by other sensors. Bacteria itself is a fermenter where it utilizes nutrients and it produces product. So it is micro fermenter, I can say. So in as in the second part of the question, human digestive system. Human digestive system has so many useful bacteria. I can say namely lactobacillus, other even E. coli as well. So they will utilize the nutrients what we are giving into the stomach, and that is external atmosphere for that micro fermenter, what I can say bacteria. So they will take this nutrient inside of their fermentation system, their body, their cellular body, and they will generate, like they will utilize that nutrients as their source of carbon and nitrogen, and they will generate a product. So that product will be either released or it can be utilized within the cell. So it is nothing but like same. We are giving nutrients to the industrial fermenter. We are taking product and we are utilizing. Suppose if it is vitamins, suppose vitamin C or vitamin D, something we are produced from the industrial product. Again, that vitamin can be utilized for the consumption, like wherever it is needed. Same vitamin C can be produced by the microorganisms within the stomach, like within the human digestive system. So yes, I think it is answered.
Yeah, very true, because most of the probiotics which are now into market, so they are trying to aim to improve our immune system. Yeah, we also have that overuse of antibiotics is going to bring down the vitamin D levels. So it is related. Yeah. So
like I can say, like previously the food which was taken contains everything in it, and the bacteria which was present in like a probiotic which was by default present in the digestive system might have generated sufficient D vitamins for the people. But whereas in recent trends, people are becoming a narrow, like selective food takers. So either they are vegetarians or they are exclusive non-vegetarians, or they may interested in taking sweets, or like they are not taking a complete food. So the microbial flora present in the digestive system obviously get disturbed, and the supplement has to be given externally.
Yes. So there is one more question from Mohini, that what are the challenges in terms of growing microorganisms in extreme conditions in fermentation? So what I wanted to know is how to overcome challenges in growing thermophilic, halophilic organisms that may have a negative effect on large-scale fermentation.
That's what I want to mention in that slide. Like at present, the organisms which are used in industries for the production of specific products are very stringent. Like we can't play with them at extensive atmospheric conditions. So only way what we can do is, like, we have to adapt the microorganisms, or we have to look into the recombination technologies. We have to select the host cell which can be adapted to the extreme conditions, and parallelly produce equivalent product that our E. coli is producing in stringent conditions. So like suppose we are using 37 degrees for E. coli, it may be like 1 or 2 degrees here and there it can survive, but if we use same like 45 degrees for the same E. coli, it may not survive, and we may not get that specific product. So if we take extremophils, methanogens are extremophils which can survive at high extreme temperatures, and we can do this recombinant genetic engineering in that extremophils, making sure like it should express that specific protein. With we can do that, but the chances are I don't know, like the equal chances may not be same for both the organisms. It has to be like developed. It has to be done in future.
Yes. So there is one more. So it is difficult that we are not able to tap the full potential of the extremophiles.
Yes, that's what.
Yeah. So one from Akila, that how to prevent the formation of inclusion bodies during bacterial fermentation upstream processing.
Yeah, like inclusion bodies specifically like meant for when we give some extreme stress to the bacteria, they will form inclusions. So if you want to reduce the formation of inclusion body, you have to maintain the physical parameters very strictly. Suppose pH, temperature, whatever, like nutrients, all those things should be maintained very stringently. Like suppose if you want to you have to maintain 37 degrees, means you have to strictly maintain the temperature without altering not more than 1 degree. So if instead of 37 degrees, you went for like 42 degrees, no, obviously it form inclusion bodies. Or if you come down to 25, it may or may not form. Like the parameters play a major role in forming inclusion bodies. So as well as nutrients. If it is we need to maintain around 5% of carbon source in the nutrient media, we have to strictly maintain on that thing. So if we like that carbon source has been consumed very rapidly and we are not feeding from external source with the sufficient carbon source, then obviously it will form inclusion bodies.
Yeah, so thank you, sir. I think you got your next few questions which you had put in the last slide that to add to your questions. Your last slide, you had asked for a few questions. Yeah. So
I didn't able to see. Just let me know if you can read it. I would
in your slide. No, in your slideshow, you had put in that just the audience should add a few questions.
Yeah,
so they did add a few questions.
Okay,
Yeah.
that's nice. Yeah, like only once questions are posting, then only our mind will activate and we will think beyond the boundaries and orbit next can be achieved.
Yes, sir. So yeah, so thank you. So I would like to ultimately thank you for taking out the time actually at a short notice and accepting our invitation. And you have delivered a very beautiful webinar on fermentation. So you have showed us all the different types of the fermentation, including that the economical use of various substrates, the sterilization techniques, the types of organisms, right? And also different fields in which the fermentation can be used. So these are some things that you have highlighted to our audience. I hope they take into, and hopefully if we have some who are aspiring to join into the fermentation industry, so we will be in touch with you. So
definitely, yeah. I'm always approachable and can do my best to support people who are willing to join in fermentation. But it may not be exactly in my office, but I will definitely extend my help to give information wherever things are available to explore. So and once again, thank each and everyone who are participated in the call and listen patiently all my things and took a chance to it is second time I invited by Swaruparan madam. Previously, I think it may be 1 year back or just before this corona first pandemic.
Yeah, we had tried,
so
but
this time I thought of I should not miss, though it is a short notice. I thought of prepare it my best.
So kind of you. So yeah, thank you so much, sir. Thank you for even patiently taking all the questions from the audience. So yeah, thank you so much. We are very happy to have you.
Thank you all.
Yes, sir. Thank you. So going ahead with the webinar. So once this is done, so we can now move ahead with the next segment. So as previously mentioned, Microbiology Art Competition was actually conducted. That was held from 14th to 16th September. So it's time to announce the winners, right? So the theme of the competition was impact of microorganisms on mankind. And actually, we are actually great that Professor Deshmukh sir, who is the president of the Microbiology Society India, is here with us today. Sir has been very supportive in all our initiatives, and yes, the society has also partly sponsored for the cash prize of the art competition. So if sir is available, yes, sir. Can you just speak a few words, sir, because today is International Microorganism Day. So just share your thoughts with our audience today. So over to you, sir. Sir, you're not audible, sir. You need to unmute.
Thank you.
Yes, sir. Yeah.
Thank you, Dr. Sumti.
Yes, sir.
And Dr. Swaruparani also for arranging this beautiful event. I was listening Dr. Prasad from half an hour back. He delivered an excellent lecture, which is really informative to the young generation. And his work is, or his lecture was from his experience, not only theoretical. So I really congratulate you first, rather than Dr. Prasad, because Dr. Prasad, you called him. And because of you, he is here today. Really, I'm thankful to Dr. Prasad also for accepting the invitation. And Microbiologist Society, as a president of Microbiologist Society, it is my duty to promote such activity, such beautiful activity in the subjects. And this team of Women College, they are doing excellent in microbiology on occasion of the Microorganism Day. And today again, publicly, I want to assure that we are always with this college, with this team, morally as well as financially. Because we want only the teachers. Actually, I'm always telling, I'm doing nothing. All these young teachers are doing many activities. I'm only supporting them. I'm only encouraging them. I'm happy to have a crowd of such devoted teachers around me. So I don't take much time. I'm thankful to you for giving me an opportunity to share a few words, to share my few views. And also I expect in the future same activities from your department, from your college. Thank you. Thank you very much.
Yeah,
Thank you, sir. Thank you, Deshmukh sir.
Thank you so much,
I
sir.
think I can remember a few moments where I met you in Pune during time of like some conference around 2015 or 16 when I was a research scholar at that time.
Okay. Very nice.
So,
Keep in touch.
yeah, definitely, sir.
Yes, and share such type of thoughts again in this college, and I will attend definitely.
Thank you, sir. Definitely will.
Okay.
Yeah, so thank you so much, Deshmukh sir. Please do be in support to us always. And we'll just say, give away the winners of the art competition. So I think first prize, it goes to Lalita Siani, who's from RYKS College. She's from Nashik, Maharashtra.
Yeah, very excellent college. Very good college.
Yeah, so she'll be receiving a cash prize of 1,000 rupees. So once the webinar is done with, we will be doing the online transfer to the winners. So first prize goes to her. I think there is some problem with the sharing. Yes, so just we would like to share, but
You want to share her certificate?
no, we have her artwork, so that we are trying to share. 1 minute, sir. There is some technical glitch, I think. 1 second, sir. 1 second. Okay, now it's visible, sir?
Yeah.
Yeah, so the first prize, it goes to Lalita Siani. Yeah, she's from Nashik, Maharashtra. जा नहीं रहा है. So 1 second. Yeah. Yeah, she's a final year student, and this is her artwork. So she has shown both the positive and the negative side. So impact of microorganisms on mankind was the topic. The second prize goes to Nargis Jamil. So St. Xavier's College, Ranchi. So she receives a cash prize of 750 rupees. So this is her art.
Yeah, Sumati, sorry to interrupt.
Yeah.
Are you sharing the screen?
Yeah, I am. Is it visible to everybody?
No, madam.
No.
No.
Okay, I thought it is visible. 1 minute.
Screen is visible, but yes.
Okay, I have to redo this. Okay. So this is of the second prize. Is it
Correct.
visible?
Yes, it is visible.
Okay.
Excellent.
1 minute. I'll just redo the whole thing because first one we missed out. Okay. There is always some technical issue in all this online.
Yes.
It's visible now. Everything is visible now.
Screen is visible. Files are visible, not the picture.
Oh, what's wrong with this?
You have to click on art competition.
Yes, sir. Yeah. Now it should be visible. Now?
Only first winner is visible.
Okay. Right.
Correct. Very good.
So this is of the first prize winner, Lalita Siani, right? So she has shown the global impact of COVID and also the positive effects that microorganisms have on, say, pollution treatment, production of antibiotics. So she's stuck to the theme. Second prize, Nargis Jamil from Ranchi. So this is her art, overall depicting the effect the microorganisms have on mankind. So be it the positive effect, fermented food production, and we have even the negative effect causing disease in plants, humans. And the third prize, it goes to Aishwarya Nair. She's from Bhaskaracharya College of Applied Sciences. So she has also given her positive impact of microorganisms on mankind. Just focus more on the positive effects. So because we received around 200 artworks, so we thought difficult to choose the winner, and we had to go for a consolation prize. So the first consolation prize goes to Rani, who's from our own college. So she has actually depicted the discoverer of microorganisms, also depicting the positive as well as the negative effects of the microorganism. So, and the next 2 consolation prizes were given. So these 2 consolation prizes each for 200 rupees each. So this is the second one. So this is by Srivalli from Bongir College, which is an armed forces college for women by the Telangana government. So these are the few artworks which we are sharing with you now. So these are just which were shortlisted. So a few of the shortlisted artworks, I'm just sharing it to you. So receiving around 200 and screening was quite a job. So very beautiful concepts that the students and audience have come up with. So probiotics. So many focused on COVID because it's the hot topic now. So without microorganisms, we can't survive, but they also have the potential to actually, wipe out the entire mankind if they can cause such a pandemic and huge loss of life. So this is a very unusual concept that we have the coronavirus looking into humans under the microscope. So very nicely done by most of them. Yeah, so that, yes. So there we end it because these are the winners. So the 3 winners and the consolation winners, we will be contacting them after the webinar and we'll be transferring the money online. Right, sir. So I now request.
Madam.
Yes, sir.
Madam.
Yes, sir.
There are 200 entries as per you have told.
Yes, sir.
Correct?
Yes.
And we are selecting 3 + 2, 5 students.
Yes.
So my request is that if you can select 5 more, we'll sponsor. Don't worry about the money.
Okay. Sure, sir. We will do
So
that.
five students give consolation prizes.
Sure, sir.
And because entry number is so huge.
Yes, sir.
And particularly this picture which you have shown last picture.
Coronavirus and looking.
Coronavirus is watching human being through microscope.
Yes, true.
Really wonderful brain of the student.
Yes, sir. Yes.
I want to appreciate that student also.
Yeah, that was, I think, P. Sonia Gandhi. I don't remember the college.
Very good. And you got a response right from Maharashtra, Delhi, from
Yes.
Hyderabad, and from other places also.
Yes, all far-reaching responses we got. So
Yes,
thanks. Thanks to you also. For promoting the event and being hand in hand with us through the financial aid. Yeah, thank you, sir.
Okay.
So
Thanks, Sumati. Out of curiosity, one question, let me know, how many members joined at the end of, till the maximum numbers. But do we have any count?
I think Swarupa ma'am will let you know, sir. Once the webinar is done, we'll be sending you the details.
Okay. Just for the thing, how many have attended.
Sure, sir. We will share the details.
Okay. Thank you.
So over to Swarupa ma'am. I think she can present the vote of thanks.
Yeah, thank you, Sumati. So I would like to conclude today's session by thanking our beloved secretary, Mr. Ronald Rose, IAS, for extending his constant support. I also render my gratitude to Dr. Praveen Mamidala, JS, for being constant motivator towards research and development. I also thank our JETSIVA ma'am, Dr. Rajni Paspuleti, for encouraging us. And my sincere thanks to our principal ma'am, Dr. G. Nirupa, for extending constant support in making this webinar a great success. I extend my heartfelt thanks to the staff, students, and all the enthusiastic participants who made this event successful. And I also thank our today's chief speaker for enlightening us on the fermentation technology. And finally, my special thanks to Professor A.M. Deshmukh sir, President MSI, for his support and sponsorship for the art competition. We will contact the winners soon for sending the cash prize. And thank you again for being with us today. Have a great evening. Thank you, sir. Sir, we will end this session today, sir. Yeah, you can leave the meeting. And also we have shared feedback link. Please fill the feedback link for getting the certificates.